So yesterday we talked about how to name the functional groups, and I told you that you're going to see that we're going to be using each of those different functional groups as we go through the organic compounds of life. So today, the first thing I want to go over with you is how do we put these things together. So the base organic compound that everything kind of starts from will be just this carbon chain. And the little bitty pieces of the carbon chain are called monomers, mono for one. So what do you think we call it when we put a long chain together? Polymers. So the idea is that every different one that we're going to talk about, carbohydrates, lipids, proteins, nucleic acids, they all have basic building blocks. The building blocks of a protein are the monomers of the protein. In the case of protein, the monomers are amino acids. In sugars, the monomers are the monosaccharides, and then we build them into more complicated things. So each different class of compounds has its own monomers. When we put them together, we form polymers or chains, which then fold up and have specific shapes and have specific jobs. So this was a question in the pre-lab a lot of people asked me about yesterday, about what is it, how do you draw a synthesis reaction? And actually it's called a dehydration synthesis reaction. What does it mean if you get dehydrated?
Your body doesn't have water.
Right, your body doesn't have enough water. Like maybe you sweat too much, you haven't drank enough. So the name of the reaction that puts these monomers together is actually called a dehydration synthesis reaction. And the reason why, if we look at it, is that you're going to have something, and in the lab you may have drawn it more complicated than this, but you have something that has, let's just say it has an OH sticking off of it. And then you have some other thing that also has an OH sticking off of it. And what basically ends up happening in a dehydration synthesis reaction is that the H from one of these and the OH from the other are going to combine and leave as water. And what's going to be left will be this and this now chained together. So it's called a dehydration synthesis because this sort of becomes dehydrated. It loses water in order to hook together. Now in your notes you're going to see one there that's more complicated looking. It's got these hexagon shapes and shows like an OH here and an OH here. It's the same thing. It could be any two things. It could be two amino acids, two fatty acids. It could be anything hooking together. It's always going to work the same way. It's going to be the H from one, the OH from the other. Those are going to get together, and then this is going to end up being a bond between the two. So that's a dehydration synthesis reaction or a condensation reaction. The opposite of this is a hydrolysis reaction. Hydro refers to water. And you may remember from biology, I don't know if you remember this, but to lyse, when a cell lyses, it means it explodes or it breaks. Like in a hypertonic, hypotonic solution, you learn about how the cell swells up, and if the cell keeps swelling, it would explode, and that's called lysis. So hydrolysis would mean the breaking using water. So it's the opposite. So for example, your body has to build proteins. Your muscle is made out of protein. You take amino acids, and your body uses protein synthesis, and it takes the amino acids and uses these dehydration synthesis reaction to make a big long chain. And those big long chains become your enzymes, the hemoglobin of your blood, your muscle, all the different kinds of proteins that were used in our body. But on the flip side, let's say you eat a steak. Well, the steak doesn't just turn into muscle. You have to digest it. So you're doing the opposite. The enzymes in your stomach and in your intestines are doing hydrolysis reactions. They're taking a protein or a complex carbohydrate or whatever it is, and they're breaking it into pieces. So these two reactions, the condensation and the hydrolysis, are opposites of each other, and they are basically the two reactions that build and break down all the organic compounds that we're going to be talking about. Okay. Here's a diagram. This is the one that's in your notes. And like I said, if you drew this one in the lab, that's fine. It's not necessary. It doesn't have to be this drawing. Because the important part of what's going on is not this. The important part of what's going on is that you have two things, and that the OH on one of them and the H on the other are going to hook together and leave. That's the important part of the reaction. So whether you drew this complicated one, or whether in the lab you just draw a circle and an OH and a circle and an OH and showed a little dotted line around that, you've still represented the same thing, just so you know. That's the important part of the reaction. Okay, so isomers. Probably never learned about these. I think even in chemistry, I don't know if you talk much about it. In AP even, I don't know if you ever talk about isomers. Basically, the thing is, organic compounds, a lot of them, have the same exact formula, but then when you draw them, you can draw them in more than one way. If I gave everybody, I don't know if you ever played with the little molecular model units with the little balls of oxygen and hydrogen, and you build stuff with it. I could give everybody 6 carbons and 12 hydrogens and 6 oxygens and say, build me something. And I would probably get, there's only certain ways you can do it. Like hydrogen can only connect to one thing, so hydrogens would have to be on the ends somewhere. But the bottom line is you might come up with a whole bunch of different ways to put those together. Those are called isomers. The reason it's important is that the way that they are arranged in space three-dimensionally changes the way they react in your body. And so here's a very good example. Well, this is not a very good example. This is just 2 isomers. It's hard to really see how they're isomers of each other, but basically they're three-dimensionally, they're arranged differently. But here's a practical application of it. Back in, I want to say the 60s, there was a drug called thalidomide, and thalidomide was used in pregnant women. It was used to control morning sickness. You get all nauseous and they would throw up and everything. And thalidomide sort of was it had a calming effect and it stopped the vomiting or whatever. So it became widely prescribed. What they didn't realize at the time was that thalidomide has 2 isomers that are mirror images of each other. They look almost the same, but it's sort of like trying to put your left shoe on your right foot. It doesn't quite fit right. And it turns out that the body breaks down the one form of thalidomide and uses it as a sedative, but the other form of thalidomide caused this. It caused mutations in the developing children. And so there was this widespread number of children across the nation that were born with this same genetic or developmental defect with these very, very short arms. In fact, it was so common that if you know that Billy Joel song, We Didn't Start the Fire, there's a line in that song that says children of thalidomide. I mean, it was enough of a big deal that when he's talking about Eisenhower and all these different things, he mentions this. Obviously, they don't prescribe it anymore. But that's a good example of a practical application of isomers. Not every isomer is going to behave the same. Just because it has the same formula doesn't mean it's arranged the same way, and doesn't mean your body will use it the same way. So the first one that we're going to talk about today is carbohydrates. The building blocks of carbohydrates are going to be your monosaccharides. This is from the lab. If you worked on the pre-lab, you'll know that carbohydrate is basically, the meaning of it is carbon water. And if you look at the formula, that makes sense. Because basically the formula for a carbohydrate is just a carbon and a water. What's the N represent? Somebody told me normal. I had all kinds of nitrogen. No, and it's not nitrogen either. It's a variable. For example, in glucose, N is equal to 6. That means our formula would be for glucose C6H12, 6 times 2, O6. Now, in ribose, like RNA, DNA, RNA, ribose, it turns out that N is 5. So the formula for ribose would be C5H10O5. So this is just basically what they're saying is even complex carbohydrates are basically just a multiple of this. I don't know if you remember in chemistry learning empirical formulas and molecular formulas. This is basically the general empirical formula for every carbohydrate. They all consist of this ratio, kind of a 1 to 2 to 1 ratio. And then the actual formula will be a multiple of that. As you see real complex ones, the ratio kind of goes off a little bit, but it's still going to generally look like this. A lipid, on the other hand, is also made of carbon, hydrogen, and oxygen, but a formula for a lipid would be something like, let's say, C24H48O3. It's hardly any oxygens and all carbons and hydrogens. But if you saw C24H48O23, that would be a carbohydrate. Even though it's off by 1, it's generally a 1 to 2 to 1 ratio. So that's what you're looking for in carbohydrates, and you'll see when we talk about lipids, that's the last one we're going to talk about today. Okay, so our building blocks are monosaccharides. which are single sugars. When we build a bunch of them, we get polysaccharides, which are complex sugars. Glucose is probably the most common monosaccharide. Everybody's heard of glucose, I hope. It's the one that's floating through your blood. It's the one your cells are using for energy on a general basis. Your blood glucose levels pretty much stay the same. You maintain a homeostasis. When you eat more food and you make more glucose, you take it away and store it. But then when you haven't eaten in a while, you bring out more glucose, and then your cells have it to use, or when you're exercising or whatever. The main job of monosaccharides, they're used for energy. Ribose and deoxyribose, I just mentioned, 5 carbon sugars. They're part of DNA and RNA. Couple of disaccharides that are common. Sucrose, which is like table sugar, the kind that you use for baking, or the kind that you might put in like coffee. That's sucrose. It's a disaccharide. And lactose is another disaccharide. Now here's another example of isomers. Sucrose and lactose are pretty much isomers of each other. They have the same general formula. But I've never heard of somebody who couldn't eat sucrose. Pretty much everybody can eat sugar, sucrose sugar. I have heard of people that can't eat lactose though, because lactose is arranged differently. And some people, in fact a lot of people, don't have the proper enzyme. It doesn't function well, or it doesn't function at all, and they're lactose intolerant. So when they eat lactose, they get nauseous, or they get an upset stomach, or they get diarrhea, because they don't have the proper enzyme to break down lactose. But yet they can break down sucrose, which has the same formula. So this would be another example of where isomers have a different structure, even though their formula is the same. I'm going to show you a diagram on the next page, and I'll probably later give you a handout that's just got sample diagrams of everything. So don't worry about if you're trying to scribble down what everything looks like. This is some pictures of monosaccharides. They can sometimes be drawn like this, like as a hexagon or a pentagon, or they can sometimes be drawn in a straight line. Here's the dehydration synthesis reaction. This would actually be sucrose being formed from glucose and fructose. But it's just showing you the building blocks, the monomers, and then in that case a dimer. Polysaccharides, multiple sugars. Plants store their energy as starch. So when you think of things that are starchy, foods that are starchy, it's typically all stuff that comes from plants. Potatoes, rice, wheat, oats, corn. These are basically ways that plants store their food, and then we can take the starch from those things and we can then manufacture products that we want to have. So that's starch. When I ask how animals store their energy, the most common answer I get is fat. And that's true, we do store energy as fat for long term. Like if you eat way too much all the time, you're going to store your energy as fat. But what keeps your blood sugar stable between breakfast and lunch, or between when you went to sleep last night and when you got up this morning, is what's called glycogen. Glycogen is the main way you store energy. You store it in your liver. It's sort of like having a nice little IV drip, so that as you go throughout your day, it just keeps slowly releasing sugar as you need it. Now you eat a meal, it picks up the sugar and holds on to it. And so glycogen, there's a hormone that breaks it down. Well, insulin's one of them, and there's another one called glucagon. But you basically can release or store up glycogen. Now, if you want to lose weight, burning glycogen isn't the way to go. You're going to cut your calories and exercise so that you go past the glycogen, you use that up, and now you're going to go to fat as your next way of getting energy. Cellulose and chitin, if you remember chitin, we were talking about chitin, and I told you it was chitin. Those are actually structural carbohydrates. As a matter of fact, another good example of an isomer. You can digest starch, you can digest glycogen. You can't digest cellulose. If you don't believe me, then when you're done floating paper clips and pricking your finger to check your blood and doing belly flops to see if it really hurts, you can then eat some corn, which is covered, all the little kernels are covered in cellulose. But don't chew it. Just swallow it. And then wait a few days, and you will see that you cannot digest cellulose, because that corn will come out looking just the same way that it went in. So, don't do that. Oh, well, you could wash it off, and then you'd have corn again. I mean, it's just the gift that keeps on giving. I'm kidding. I know, that was too much, too far, a little too much. All right, and you don't have to know peptidoglycan, but that's yet another different carbohydrate that's used in bacteria. So these are just different forms. These are polysaccharides. You have your monosaccharides, which are your simple sugars, disaccharides where you put two together, and then polysaccharides where you have a whole bunch of them put together.